All right, our next speaker is Anshul Kundaje, who's an associate professor of genetics and computer science at Stanford. He develops cool statistical and machine learning methods for large-scale computational genomics. Besides all the cool work he does, he's also worked on large-scale consortium projects while a postdoc at Stanford and at MIT, specifically the ENCODE project and Roadmap Epigenomics project. He's the recipient of the 2016 NIH Director's New Innovator Award, as well as the 2014 Alfred Sloan Fellow. Welcome, Anshul.
Okay, can you hear me? Awesome, great. So first I want to just start by thanking the organizers for this kind invitation. It's also good to be back at Columbia. I actually did my PhD here many years ago. And I happen to coincidentally live in Palo Alto on a street called Columbia Street. So, there's a lot of nostalgia coming back. Anyway, today I'm going to talk to you about what my lab works on. We work on machine learning methods to decode genomes. So it's a little bit out of distribution from the classic MLHC talks, but hopefully it'll be of interest because genetic data and molecular data is becoming a key component of, I think, the general healthcare ecosystem. And so hopefully I'll show you how we can use methods and models to understand genomes and potentially use that to decipher genetic basis of disease. All right, so before I start, I just want to put the genome in context because when you're decoding genomes, it's not as simple as looking at a piece of DNA. The same genome is present in every cell type in a person's body. It has the exact same copy of inherited genetic variation. And so when you're interpreting genomes, you're trying to understand what genomes encode, you really have to interpret them in the context of the thousands of potential cell types and cell states that exist in the body, right? So having put that in your mind, let's sort of dive deeper into what the genome is made of. What really defines a cell's state is really, in some sense, the repertoire of genes that are expressed. There are about 26,000 genes in the genome. And each cell type, as I mentioned, has a specific combination of genes that are expressed, and they form intricate gene programs. And so if you think about, there must be a control system that regulates gene activity, and that control system also happens to be encoded in the DNA itself. So you have these, the yellow and orange, what are called regulatory control elements, and they are pieces of DNA that basically activate or repress genes. And there are millions of these elements in the genome. It's only recently that we've been able to map a large proportion of them. Many of them are still mysterious. We don't know exactly what they do. And just like genes have very cell type specific activity, these control elements also have very cell type specific activity, right? And if you think about what they're made of, these control elements, they encode sort of an orthogonal language. Everyone knows about, the classic coding sequences of genes, right? They have codons, and those codons code for proteins. But the regulatory elements also have their own language. They have syntax, they have grammar, and they have these DNA words called sequence motifs. And DNA binding proteins recognize the sequence motifs. They bind in protein complexes to these elements, and then they trigger specific downstream regulatory activity. So why is this actually relevant? It sounds like a classic molecular biology, deep dive into how the cell works. Well, the reason it's important is because we can now sequence patient genomes very cheaply, or even genomes of generally healthy individuals, and you end up with millions of genetic variants, mutations, mismatches or insertions and deletions in the genome. And then if you try to figure out what these are doing, and you see where they lie in the genome, actually less than 5% of scanned genetic variation in large repositories directly disrupt protein coding sequence, right? So 95% of this genetic variation that either does or doesn't do anything happens to lie outside genes. And a large proportion of these, in fact, happen to be enriched in these regulatory control elements. So we understand and we decode the sequence features and how sequence causally drives these regulatory elements. We may be able to actually do a much better job understanding the molecular function of genetic variation. So that's the premise of this whole talk. And so let's talk about how we can decode regulatory DNA. So as I mentioned before, there are millions of these elements scattered all over the genome, often quite far away from genes that regulate. And so you first need a way to potentially scan these and find where they are. Luckily, we've had a big revolution in molecular sequencing technology. And so what we can do is use sequencing tech to profile molecular entities that tag these regulatory elements, right? So I'm showing you on the right, each of those weird little words, those are transcription factors. Those are DNA binding proteins, genes that produce proteins that bind specific regulatory elements. Each track there is the output of a sequencing experiment, and the sequencing experiment gives you a readout at every position in the genome, and the signal level at that readout, in some sense, captures the probability of a protein binding that site. So you see these spikes of signal across a piece of the genome, and you can see different spikes for different proteins, and you can see different spikes for different proteins aligning to the same position, indicating extremely intricate and complex combinatorial binding of these proteins at specific elements. And these very same proteins can have very different binding landscapes in different cell types, right? So even though it's the same protein, it has a very different binding landscape, activating a very different set, activating or repressing a very different set of elements in the thousands of cell types and tissues. So what we want to really do is to be able to take one of these experiments. We can do these quite routinely now, and we, the introduction mentioned, we've been working with several large consortia that do this at scale. So you can do tens of thousands of these experiments in hundreds and thousands of cell types, and now at single cell resolution, right? Not even in bulk. So you have millions and millions of cells with these kinds of readouts. So imagine you have this kind of a readout in the genome. I'm going to talk specifically about two kinds of experiments, ATAC-seq and DNA-seq. What they give you is, again, a readout at every position in the genome of the likelihood of some protein binding that site, okay? And it's one experiment that gives you a genome-wide readout. Now, given this experiment, we'd love to know how is the DNA sequence causally affecting that, right? Somehow the DNA is encoding that information. It's all in the DNA. So if we were able to decode the DNA and figure out the syntax that causally drives that signal in that cell type, we can potentially predict what mutations to the DNA would affect molecular function, thereby having downstream effects on cellular phenotypes, which will eventually have effects on organismal phenotype, right? All right, so let's see how we can do this. So we'll transform this into a machine learning task. Very simple, supervised machine learning problem. You have a readout over the genome. Every position, as I mentioned, has a signal from the experiment. It's actually a discrete count from sequencing experiments. You have one read or two reads or 10,000 reads at any position in the genome, so a discrete non-zero count. And what we can do is take the genome and bin it into little chunks, let's say 2,000 base pair chunks. The genome is 3 billion base pairs, so you get millions and millions of little sequences. Each of those sequences is associated with a readout from the experiment, right? And so you have a nice pair of inputs and outputs. You have sequences as inputs, little chunks of sequence, and readouts from the experiment as outputs. It's a very simple supervised machine learning formulation, and so your goal is to map sequence to molecular activity, and you can fit any model you like, right? So there's tons and tons of work in this space for decades. I'm going to talk about a slightly different version of this. In most of the current work that's been done in this space, the mapping is from sequences of some finite width to scalar outputs. And these scalar outputs are either continuous or binary. You can binarize the data and say, I see a significant signal or a non-significant signal, or you just summarize all the signal over the sequence, and you capture a single scalar value capturing the activity of that sequence. But if you actually take these signals that you get across the genome, and you really zoom at super high resolution and see what's happening at every single base pair, there's really amazing biophysics happening, and the molecular experiment is reflecting a lot of that biophysics. So what's happening, for example, is let's zoom into that particular spike of signal. You see, if you look at the reads at the coverage, the number of reads, sequencing reads mapping to each position in that little piece of the genome, you see something interesting happening. Where the protein is bound, you see something called a footprint. Basically, the protein is protecting the DNA from the enzyme that is used to digest and create the reads. And so you get a little valley of signal. And right next to that valley of signal, you see these really nice, strong spikes. And the precise shape and the contour of the profile at single base pair resolution mimics the precise protein-DNA contacts. The way the protein domains contact DNA in 3D space is beautifully reflected at single base pair resolution. If you summarize this data into one scalar, you have lost all that beautiful information. So why have people not modeled the data this way? Because it is very sparse and noisy. You have like 1 read, 2 reads, 0 reads. There's lots of dropout, right? So you typically aggregate, so you get statistical power, and then you can do machine learning, right? So we thought, let's try to really not lose resolution and try to model this data in its rawest form at single base pair resolution. We didn't even know it was possible, but let's try. So here's the model that we have. This is based on a previous model we published in 2021, where we, for the first time, showed that this is in fact possible. You can map DNA sequences to these molecular readouts at single base pair resolution with super high accuracy. But we've done it for a much simpler experiment, protein DNA binding experiments. We're now going to do this for a more complex experiment called a chromatin accessibility experiment using the DNA-seq or ATAC-seq enzymes, okay? So the way this model works again is your goal is to learn mappings from 2,000 base pair DNA sequences to 2,000 base pair profiles of discrete counts, right? That's the mapping function that you're trying to learn. You use a simple convolutional neural network. The input to the model happens to be a one-hot encoding. So you have 4 channels, ACGT, over 2,000 base pairs. You have a layer of convolutional filters. The convolutional filters are trying to learn subtle patterns, sequence patterns in the DNA. We have multiple layers of dilated convolutions. These dilated convolutions are again trying to learn higher order patterns in an efficient way without leveraging too many parameters. We have residual connections. And then the key thing is that the model has 2 outputs. One output is very similar to what people do before. It is a single scalar. It's capturing the total signal over the entire sequence, the total number of reads, okay? And there's a second output from the model, and that second output is trying to take the total number of reads and distribute them at every position in the output sequence, right? So we have 2 loss functions. One loss function is like a Poisson loss, which is trying to capture the noise in the, or model the variance of the total counts of reads over the entire sequence. And then we have a multinomial negative log likelihood. A multinomial distribution captures quite nicely the likelihood of observing reads at every position in the sequence. So it's a very simple joint loss function, a very simple convolutional neural network. And when we apply this and train this across the entire genome on a subset of chromosomes, so we hold out a subset of chromosomes and we train on a subset and we try to see how the model predicts on the remaining subset. So if you look at some predictions, I'm going to sort of show you examples and then show you global performance statistics. It's always good to look at the data, and so I'm going to show you a lot of data. So this is the ATAC-seq experiment that actually generates the reads. We are zooming into a small 300 base pair region of the genome right next to the start of a gene. This is the observed data at single base pair resolution. This is the data predicted from sequence alone on a held out chromosome. You can see that they are almost indistinguishable, right? It's very, very accurate. Now, what's really interesting is this experiment called DNA-seq is also trying to capture the exact same phenomenon using a different enzyme. So the only difference is the enzyme that's used to target the DNA. The ATAC-seq experiment uses something called a TN5 transposase, and the DNA-seq experiment uses an enzyme called DNase, right? So they're technically capturing the same biology, but if you look at the actual signal readout, they look very different, right? And you can see the model that's trained on the ATAC-seq data captures the ATAC-seq signal distribution beautifully, and a model that's trained on DNA-seq captures the DNA-seq signal distribution beautifully. So the model is able to mimic, is able to learn from sequence alone exactly what's driving the profiles at single base pair resolution, and yet there is a strange difference between the 2 experiments. They're supposed to be capturing the exact same biological phenomenon. So immediately, the thing that you should think in your mind is, even though my model looks almost perfectly accurate, something is wonky, right? And the wonky thing is that the experimental readout from the experiment is the biology confounded with technical artifacts of the enzyme being used, okay? So how do we know this? Well, okay, so we can do some of the, this is one cherry-picked example, but we can actually, do performance plots across the entire genome. We get pretty nice high correlation in the total read counts or bins over the genome. And if we look at the shape of the profile, as I mentioned before, we have a multinomial distribution over 1,000 base pairs. We can compare the predicted distribution with the observed distribution using a Jensen-Shannon divergence. This is the upper bound. If you took replicate experiments, what would that look like? This is the distribution. This is the observed versus predicted. And these are sort of negative controls. So if you just randomize the profiles and you compare them to the true data, this would be the lower bound, this would be the upper bound. We are quite overlapping the upper bound, which visually, you saw in the previous slide that the predictions really look like the observed data. And so globally, we see very high performance, right? So this is pretty state of the art. We're getting very high performance. But as I mentioned, something looked off. And you can see that. You can actually do a lot by interpreting your model. So we do a lot of feature attribution using classic sequence attribution methods. We had built one called DeepLift in 2017, published in ICML. It's an extension of saliency maps. It's just instead of using gradients, for example, for backpropping, from the output to the input and getting feature attribution, we just do a finite difference approach. And this is very similar to, Shapley values and deep Shap, for example. But the key idea is you can take the prediction of the model at any position in the genome, and you can backpropagate through the neural network and get base resolution attribution scores for every base in the input sequence. When we do this, again, you can see this is the observed data. This is the predicted data. Beautiful, right? Looks perfect. But when you actually look at the sequence attributions, you see some things that make sense, and you see some things that don't make sense. The things that make sense is right here in the middle. This particular motif you see right here, this DNA word, happens to be the binding site of a very well-known protein called CDCF. It's a well-known binding site. But then there's all this other spurious noise. It looks like noise, just a bunch of stuff jumping up and down, right? All over the sequence. And we see this all over the genome. So we wanted to actually figure out, this is for one piece of the DNA, one regulatory element. We have hundreds and thousands of these across the genome. We can try to summarize these predictive patterns across the entire genome. And so we actually have this algorithm called TF-Modisco. What TF-Modisco does is it takes all the sequences in the genome, uses the model to interpret and identify base pair resolution attribution scores, throws away pieces of the sequence that the model claims are not important, and then runs a classic community detection method to cluster these subsequences into unique patterns post-alignment, and then collapses them into non-redundant motifs. Okay? These are globally predictive patterns. When we do that for this model, we see something fascinating. So it's picking up DNA words of classical proteins that are supposed to be binding DNA and driving activity, but it is dominated by this kind of weird motif you see right here, this CAG pattern, CAG. You'll also see contamination of these motifs with lots of GCs, pathological signal all over the place. And this particular motif we are seeing here happens to exactly be the recognition sequence or the bias, the sequence recognition bias of the TN5 enzyme. So what this is telling us is that the model is not only learning some of the interesting biology that we wanted to learn, which is these binding sites, the lexicon of sequence recognition codes of all the proteins that drive the signal, but they also learn the enzyme bias that heavily infiltrates all parts of the model. Right? So this is bad, because the model looks like it's perfectly predicting the data, but it is clearly confounded. So how do we deal with this? Well, so what we did is we figured out a way to automatically correct the bias using neural networks to their best ability. So luckily what happens is if you look at this data across the genome, right, you see these spikes of signal, but these spikes of signal aren't all over the place. There are only about 200,000 spikes in 3.5 billion base pairs. And so vast majority of the genome is just background signal, okay? So technically, that background signal should just be pure noise, unpredictable from sequence, right? But if there was enzyme bias, the background should be perfectly predictable, and you should be able to learn just the enzyme bias by training some kind of sequence model to predict the background, right? Not the signal that you care about. So all we do is we take the same neural network I showed you before, and instead of training it on the signal regions that have high signal, we specifically focus it on the background. And as soon as we do that, it's able to predict the background extremely accurately. So if the background was just pure noise, right? It's just noise coming, Poisson noise from sequencing, it should not be predictable from sequence. But you can predict it almost perfectly from sequence, indicating that the background is also being driven by something in the sequence. And since there's no real molecular events happening in the background, the only thing that could be explaining it is the enzyme bias, right? And that's exactly what we see. If we again take the model that was trained on the background regions and we interpret it, we take these sequences, we use deep lift to identify contribution scores, and then we use Moodyscore to collapse them into motifs. They learn these beautiful collection of 9 motifs. These 9 motifs exactly look like the confounding motifs I showed you before. You can see this classic CAG pattern, right? And so we now have a beautiful model that has captured just the enzyme bias, and another model that has captured biology and enzyme bias confounded. So there's a pretty simple solution now. I'm sure you've all thought of it. You can literally just regress out the enzyme bias from the previous model that was confounded, and the residual should just be the biology, no bias, right? So that's exactly what we do. We just fit a simple, 2-path model. It's the exact same model as I said before. One half of it is trained on the background, the other is trained on the whole genome, but you first fit this bias model, you regress the bias from the signal you want to predict, and then you fit a separate neural network on the residual. Okay? And now I'll show you the magic it does. It's quite fascinating. So let's go back to our example. Here's the observed data. Here's the predicted data with no bias correction. Beautiful, as I showed you before, almost indistinguishable. As I showed you before, you learn the biology and then all this garbage noise. Now remember, we had the bias model, so we can just take the bias model and predict what this sequence would have generated. If you just take this piece of sequence and you push it through the bias model, what would bias alone look like? This is what the bias alone looks like. And you'll see that the bias actually looks a lot like the signal that we had before, right? Except in this region right here, you see a depletion of the signal in the actual data, and the bias model predicts lots of signal. If you interpret this sequence through the lens of the bias model, you see all the spurious bases that we saw in the original model are exactly the ones that we see in the bias model. So now when we regress the bias out and we look at this latent residual signal that is supposed to capture the true biological footprint of this protein, this is what it looks like. This is the residual. You can see a beautiful footprint right here with these nice spikes at the edge, and this was completely hiding behind the bias, right? And the model just reveals that beautiful footprint hiding underneath the data. And now once you take the residual model and you interpret it, you'll notice that there's almost no spurious noise in the background, and the only thing that pops out is the perfect CTCF motif, and it reveals 2 little repressors right here, which are completely hidden when you had the bias, right? So now we have a model that learns bias from the data, regresses out the bias. We can look at the latent underlying biological signal, and we can interpret the underlying sequence drivers of that latent signal. And this substantially outperforms previous statistical approaches for bias correction. Okay? So these are 2 other strong baselines, previous methods for trying to eliminate enzyme bias. You can see that they actually don't do a particularly good job. There's lots of residual bias in there. And so now if we use this bias-corrected data, we take the residual model, we use it to interpret sequences across the entire genome, and we relearn motifs, as I showed you before, by aggregating high importance sequences. You can see that we pick up beautiful motifs, around 80 to 90 of them. This is the lexicon of sequence motifs of all proteins driving activity in that cell type, and there's no bias remaining at all. That's really nice. So we have an automatic bias correction strategy. And just to show you how important this bias correction is, these are 3 different cell types. This is a blood cell type, this is a leukemia cell line, and this is a liver carcinoma. So they're very different cell types. These are 4 different proteins. They have different DNA recognition codes. These are the words that they like to bind to. And the interesting thing about these 3 proteins is they are very cell type specific. So this protein is expressed only in this cell type, this protein is expressed only in this cell type, and this protein is expressed only in this cell type. So what should happen is this protein should produce a footprint only in this cell type, this protein should produce a footprint only in this one, and this protein should produce a footprint only in this one. And this is a positive control. This protein is expressed in all 3 cell types, so it should have a footprint in all 3. Now, if you look at the red curves are the original uncorrected signal. And now just look at what they look like if you didn't do bias correction. You see footprints in all cell types for all proteins. These footprints that you're seeing, these red footprints, are complete artifacts. They're just artifacts of the enzyme bias. Once you do the bias correction, you get the black curves. And the black curves give you something beautiful. You can see that these completely flatline in these 2 cell types. You see a nice little footprint here. You see another footprint here with flat lines here. You see a super deep footprint right here with flat lines here. And the positive control shows a consistent footprint across the board. So this is really nice. We're able to detect these really cell type specific events, regress out the bias, and also decipher the underlying causal sequence syntax driving the signal. All right, so let's get to the second part quickly, which is now that we have these models, and this was one model trained on one data set capturing the regulatory map of elements in one cell type. Now imagine that we have thousands of these and thousands of cell types. We have trained these models on every possible cell type that we can get a hands on for data. So we have a big compendium of tens of thousands of models. Can we use these to understand which genetic variants in the genome are functional in which cell types and the underlying sequence syntax that they are disrupting, right? So again, you have these, all humans have 2 copies of the genome, and you have sometimes 2 different versions of it, right? So you have this copy, which might be benign, and this copy, when the C becomes a G due to a mutation, this particular version of the sequence might have a genetic risk towards a disease. That's what we want to figure out. So I just want to show you what this, we can use a very simple trick in silico mutagenesis. Once you have a genetic variant identified in the genome through some sequencing experiment, you can just push it through the model. You can push the original sequence with the G version. You can push the mutant version, which has an A, and the model just predicts 2 output signals, right, for the 2 different sequences. You can contrast them. So here I'm showing you one particular genetic variant. You push it through the model. You can see that one version produces this red curve, the other one produces the blue curve. You can see a pretty big difference between them, right? And you can interpret the model and see what is breaking. So if you take this version of the sequence with the G, you can see there's this DNA word with this DNA word with this one and this one and this one, right? I'm not going to go into the biological details, but it's beautiful and intricate. There's a awesome syntax, collaborative, cooperative, interactive syntax between 5 different proteins driving that particular element. And you can see once you swap that G to an A simple mutation, right, it breaks this DNA word. But you'll notice that all the other DNA words also reduced in activity, right? So what this is telling you is that the DNA words that are in the sequence are not acting independently. There's massive epistatic interactions. And so a single nucleotide mutation, even though it seems like a small change, can have propagative effects within 1,000 base pairs, but those 1,000 base pairs then interact with tons of other regions in the genome, thereby propagating the effect further. And you can see how a single base pair mutation can actually create a pretty amplified effect over the entire genomic system in that cell type, right? So it's super interpretable in some sense. First of all, we can not only predict these effects very nicely, but we can also try to identify the underlying causal sequence drivers that are being disrupted by the sequence, right? So we have mechanism underlying the disruption. So we've done large-scale evaluations, and I want to make a really important point here. You won't recognize these names if you're not in our field, but I'll just tell you what they are. So these are precision recall curves where experimentally thousands of genetic variants have been experimentally verified to have effects or no effects, right? So you have two classes of variants. One class of variants has effects. The other class of variants does not have an effect, right? So you have tens of thousands of these. And we are training our model just on the reference genome without any information about mutations. And we're going to predict in silico the effects of these mutations. And the simple precision recall curve. So random baseline based on the class imbalance would be 0.02, the area under the precision recall curve. If you take previous state-of-the-art methods from a previous generation, SVM models, sequence SVM models, that's the delta SVM model. This is the precision recall curve. DeepMind recently released a pretty large-scale, it's almost like a foundation model, a very large transformer multitask model with billions of parameters. Very, very interesting, powerful model. That's, that's the red curve. Sorry, that's the blue and the the green and the red curves is the Informer model. That's the DeepMind model. And then this is our simple CNN. Our simple CNN is a single task model with a handful of convolutional filters, and we dramatically outperform the big model, okay? And this is not just in one benchmark. This is unpublished work, by the way. We'll have the paper coming out soon, but we have taken multiple benchmarks from the Informer paper and validated against their benchmarks, and we outperform them comprehensively in all benchmarks, right? The key point I'm trying to make is that you do not necessarily need giant models trained on billions and billions of data sets. If you're careful with the way you train the models, taking into account some of the biological aspects of the experiment, you can train small models that can, we train these models in 4 hours on a single A100 GPU, and they outperform a model that was trained on, I think, several TPUs for several months, right? So that's pretty nice. We have other kinds of effects. So we cannot just predict whether a variant has an effect or not. That's a binary prediction. We can also predict the actual effect size and the direction of the effect very accurately. This is the observed effect size and direction from actual experiments, and this is the predicted effects from our model. We get a pretty nice correlation, 0.6929. This is an underestimate, by the way. I don't have too much time to get into it, but the gold standard here is a little confounded. So for example, all these values that you see here sort of on the x-axis, flatlining here, those are likely false positives in the actual data, but I'm showing you the data as we see it without any filtering, and we're still getting a really nice correlation. So we actually expect that if we do the filtering and we remove the confounders from the observed data, this number is probably going to go up to something like 0.8. Okay, so we're really able to accurately predict not just whether a variant has an effect or not in terms of significance, we're also able to predict the effect size and the direction of the effect quite accurately. So I want to show you real life in the last, I'm almost out of time, so in the last 2 slides, I'm going to show you a real life example of how we can use this to study rare disease. So as you know, congenital heart disease is one of the leading and debilitating diseases in young children. It's also a rare disease in the sense that although many, many children are affected by CHD, CHD is often caused by de novo mutations. That means the mutation that's found in that patient is an N of 1. It has never been seen before in any other person. So you cannot use any other statistical approach to borrow power from lots of other patients. You literally have an N of 1, right? So you need something like a model that is trained on biological phenomena and make in silico predictions about what the mutation can do, right? So that's what we do here. And congenital heart disease, the mutations typically affect very early developmental time points of the heart, of the fetal heart. So what we got is our collaborators are able to get human fetal hearts from very early time points, 6 weeks, 8 weeks, and 19 weeks post-conception. These are the earliest time points I think anyone's ever got this kind of data from. We get beautiful single-cell resolution data. I don't have too much time to talk about that, but using that data, we can actually decipher all the cell types in the fetal heart, and we can derive cell type-specific regulatory maps. So there are about 20 broad cell types that we find in the fetal heart. And for each of those cell types, we can deconvolve them. We can derive these beautiful activity maps of regulatory elements. We can train one model, the same cheap model on an A100 GPU in 4 hours. We can train on every single cell type from the human fetal hearts. And then we can take large cohorts of patients from the PCGC cohort and corresponding controls. And we can take all the genetic mutations that have been identified and we can push them through the model. And the model will allow us to predict which of those mutations have what kind of effects in which cell type, right? That's the key question. And so I'm just going to show you, again, not going to dive too deep into this. We published this I guess this year, yeah? So you can take a look at the paper. But here's one example of a de novo mutation in a patient. You can again see the blue curve from one of the versions of the mutation. That's the reference allele in the regular population. And then the de novo mutation, you can see, destroys this DNA word. So you can make a very specific prediction that this mutation disrupts the ETV motif in a regulatory control element next to a particular gene in arterial endothelial cells. So you get very, very specific predictions. You can use that to design downstream experiments, which is what we do. So I'm going to walk you quickly through 3 experiments that validate this result. The first experiment is a CRISPR experiment, where in arterial endothelial cells derived from pluripotent stem cells, we actually we CRISPR out that specific mutation, and the model has made a prediction about what effect it would have and on which gene. So we can actually, in genome, in those cells, measure the effect of the mutation artificially induced on gene expression. That's this curve right here. You can see a significant effect, a drop in gene expression of the target gene predicted. You can also take the piece of DNA with and without the mutation and put it in front of a reporter gene and put it in the cell. It's a different kind of experiment. That validates the effect as well. And then this is a beautiful experiment. This is a phenotypic experiment. So if you take pluripotent stem cells and you create a version of those cells with and without the mutation, and then you differentiate them into endothelial cells, the ones that have mutations have morphology issues with tube formation, right? That means the arteries, right? These are arterial endothelial cells. The mutation actually disrupts the morphology of the arteries itself. So we have validation on the effect, on the gene that we predicted, as well as effects on the phenotype that we expect to see in congenital heart disease. All right, so that's just an example. We've used the same kind of techniques to actually study also complex diseases, not just rare diseases. We've done it for Alzheimer's disease. We've done this for coronary artery disease. We've done it for neurodevelopmental disorders. So it's a very powerful approach. The key trick is to get data in the right cell type. It doesn't matter how powerful your model is. If you do not have the right data, it's not going to work, right? So the key thing is to really get data in the right system to build models that really respect, in some sense, the enzymatic aspects of how the experiment is done, account for confounding factors, build interpretation tools, and then things work out pretty well. So just to summarize, Chrome BPNet, we think is a state-of-the-art model for base resolution prediction of molecular readouts from DNA sequence alone. These models are very interpretable. We can use very simple feature attribution and aggregation techniques to, first of all, reveal cryptic biases in the data. We can use neural networks themselves to automatically learn the biases and self-correct. Chrome BPNet offers state-of-the-art enzyme bias correction. We can reveal really intricate biology, which I haven't really discussed much today. Bias-free predictive motifs, motif instances, footprints at single base per resolution. What's really nice is the model also extrapolates and can predict effects of mutations in the genome. And we've used these to study, as I showed you, de novo mutations in rare disease. A key point I want to make again is that sometimes, and in many cases, small single-task models can outcompete big multitask transformer models. And our code is open source. So if you're interested in this, you can give it a shot. And with that, again, I'll thank, these are the 2 students who really drove this forward, Anushri and Anna, alongside a bunch of help from several people. And none of this was possible without our collaborators who generated lots of beautiful data. Thanks.